The mechanical properties of the extracellular matrix are known to regulate the migration of single cells, which move faster on stiffer substrates than softer ones. But whether matrix stiffness influences the collective migration of groups of cells is unclear, because, says Joan Brugge from Harvard Medical School, previous studies on the behaviour of cell sheets have yielded conflicting data.
So because of these diverse results in the literature and the importance of collective migration during tissue morphogenesis, tissue repair, and cancer, we decided to develop a more systematic and quantitative approach to define properties of collectively migrating sheets of cells, as well as to address how the substrate stiffness can affect cells under these conditions.
To analyse collective migration in different mechanical environments, grad student Rosa Ng developed a modified wound healing assay, in which mammary epithelial cells were plated around a central plastic block on substrates with distinct rigidities.
Once the block is removed, the cells can collectively migrate into the open region of the wells. We then use time-lapse imaging to monitor the wound healing process and analyze the collective and individual cell behaviors during collective migration.
As previously found for single migrating cells, the collective movement of cell sheets was faster on stiffer substrates than on softer matrices. However, Ng et al. Wanted to analyse the behaviour of individual cells within the epithelial sheet, so they turned to software developed by Achim Besser, a postdoc in Gaudenz Danuser's laboratory.
So to study the behaviours of individual cells within the sheets, we fluorescently labelled the cell nuclei and imaged and tracked their movement during the wound healing process. From these tracks, we can calculate now a number of migration properties. We found that increasing substrate stiffness increases the migration speed, persistence, and directionality of individual cells within the sheets.
These individual cell behaviours must be coordinated to collectively move the epithelial sheet forwards. To analyse the extent of cell-cell coordination on substrates of different rigidities, Ng et al. Used two different parameters. Firstly, they determined the normalised cell pair separation distance, a measure of how far neighbouring cells move apart after 5 hours of migration. The greater the separation distance, the lower the cell-cell coordination.
The other parameter we used is called velocity correlation. In this case, we correlated the speed and magnitude of the migration velocity of one cell with those of other cells a certain radius apart from it, up to a radius of 500 microns. And we calculate this for all the cells in the migrating cell sheet. If the correlation in cell migration velocity is high or close to 1, then the cells are migrating with higher coordination. And if the velocity correlation is low or close to 0, then the cells are not migrating coordinately. So using these two parameters, we found that increasing substrate stiffness increases cell-cell coordination during collective migration. Most strikingly, we found that not only are cells migrating with higher coordination on stiffer substrate, but the coordination actually extends deeper from the wound edge into the cell sheet on stiffer substrate.
Cell coordination spread from the front to the back of the sheet as wound healing progressed. And, consistent with their increased coordination and enhanced migration, cells on stiffer substrates were more polarised than cells on softer matrices.
We measured cell polarity by monitoring the orientation of the Golgi as well as the orientation of lamellipodial protrusions. On stiff substrates, the Golgi and the protrusions were oriented predominantly towards the direction of the wound closure, even for cells greater than 500 microns from the wound edge. But in contrast, on soft substrates, protrusions were more randomly distributed the further the cells were located away from the wound edge. For Golgi, it was particularly striking that forward orientation was lost just one cell layer behind the leading edge.
Ng et al. Then began to investigate why, on stiff substrates, cells at the wound edge were more coordinated with cells further back in the epithelial sheet. Intriguingly, cells on stiffer matrices were more contractile, as indicated by the increased activation of the motor protein myosin 2.
On both soft and stiff substrates, we found a contractility gradient from the wound edge into the sheet. However, the gradient was much shallower on stiff substrates. This stimulated the hypothesis that cells at the wound edge are mechanosensitive and relay their directionality cues backwards through mechanotransduction. when we reduced myosin activity with the small molecule inhibitor blebistatin, or by knocking down myosin 2 isoforms, we were able to reduce cell migration speed on stiff substrates, as well as lower their cell-cell coordination.
Ng et al. Thought that, to coordinate cell movements, the enhanced contractility on stiff substrates might be propagated throughout the epithelial sheet via cell-cell adhesions.
So we weaken cell-cell adhesions by knocking down p-coherent or alpha-catenin, or by overexpressing a dominant negative mutant of e-coherent. And we found that the depletion of these cell-cell adhesion components substantially lowered the coordination between cells during the wound healing process. This means that the physical linkages between the cells, as mediated by the coherent adhesions, are critical for promoting coordination during the epithelial sheet migration.
Moreover, perturbing cadherin-based adhesions disrupted the organization of actomyosin fibers across the epithelial sheet, supporting the idea that these intercellular junctions transduce mechanical signals from the wound edge. Yet cell coordination appears to require a balance between the strength of cell-cell adhesions and actomyosin contractility. Mild inhibition of myosin 2 actually improved the coordination between cells with weakened cell-cell adhesions, probably because full myosin activity would tend to disrupt the remaining contacts between these cells.
So during collective migration, cells at the wound edge respond to a directional cue, in this case the presence of a wound, by polarizing towards the wound. Polarization involves the reorganization of the actomyosin network, and through forces generated by this network at the cell-cell adhesions, the polarization cue is transmitted to cells within the cell sheet. So on stiffer substrate, cells at the wound edge have larger focal adhesions and higher actomyosin contractility, which increases the mechanical coupling between the cells and promote transmission of the directional cue further into the cell sheet, leading to higher coordination during collective migration.
On softer substrates, on the other hand, or when myosin 2 is inhibited, contractility is lower, meaning that mechanical coupling and directional cue transmission are weaker, leading to lower cell coordination and slower collective migration.
So since we showed that substrate stiffness can affect the properties of collectively migrating cells, future studies on developmental processes that involve collective migration should take into account not only the molecular factors in the microenvironment, but also the mechanical properties of the microenvironment. And then also it's become increasingly apparent that during cancer progression that tumor cells can invade as a collective. And since the tumor stroma is often much stiffer than normal tissues, and tissue stiffening can promote invasion and metastasis, our study raises a question of how matrix stiffness and cellular myosin contractility may interplay with cell-cell adhesion status of the tumor cells during cancer metastasis.
So we propose in our model that the mechanical coupling between cells is important for cell-cell coordination during collective migration. We are now developing a new non-perturbing method to quantify forces at cell-cell junctions with subjunctional resolution, and we will be using this method to understand how force transmission between cells may regulate cell migration behaviors.
In the meantime, you can learn more about how substrate stiffness regulates collective cell migration in the paper by Ng et al., published in the October 29, 2012 issue of the Journal of Cell Biology.
You